Imagine a microscopic instruction manual hidden inside every cell of your body. This manual, called DNA, holds the secrets to your existence. DNA stands for deoxyribonucleic acid. It's a long, complex molecule that carries the genetic instructions for building and maintaining all living organisms. DNA is often called the blueprint of life. Just like a blueprint guides the construction of a building, DNA guides the development and function of an organism. It determines your physical traits, like eye color and height, as well as your susceptibility to certain diseases. Understanding DNA is crucial for unraveling the mysteries of life. It allows us to explore our evolutionary past, diagnose and treat diseases, and even engineer new organisms with beneficial traits. DNA's structure is as elegant as it is functional. Imagine a ladder twisted into a spiral shape, and that's the basic form of DNA, called a double helix. The sides of the ladder are made up of sugar and phosphate molecules. Connecting the two sides of the ladder are rungs made of pairs of nitrogenous bases. There are 4 types of bases: adenine, A, thymine, T, guanine, G, and cytosine, C. These bases always pair up in a specific way, A with T and G with C. This complementary base pairing is essential for DNA replication and protein synthesis. It ensures that the genetic information is accurately copied and passed on to new cells. The sequence of these base pairs along the DNA molecule forms the genetic code. Think of it as a language with only 4 letters: A, T, G, and C. Just like letters combine to form words and sentences, these bases combine in specific sequences to create genes. Genes are segments of DNA that provide instructions for building specific proteins. Proteins are the workhorses of the cell, carrying out a wide variety of functions, from transporting molecules to catalyzing chemical reactions. The order of the bases within a gene determines the order of amino acids in a protein. Each 3-base sequence, called a codon, codes for a specific amino acid. The process of using DNA to make proteins is a 2-step process called gene expression. First, in a process called transcription, the DNA sequence of a gene is copied into a messenger RNA, mRNA molecule. The mRNA molecule then travels out of the nucleus and into the cytoplasm of the cell, where it encounters ribosomes. Ribosomes are the protein synthesis machinery of the cell. In a process called translation, the ribosome reads the mRNA sequence and uses it to assemble amino acids into a protein chain. The specific sequence of amino acids determines the protein's structure and function. Section 5. Inheritance, passing on the blueprint. DNA is the hereditary material that is passed down from parents to offspring. During sexual reproduction, each parent contributes 1 chromosome from each pair to their offspring. Genes located on these chromosomes carry the instructions for traits that are passed from one generation to the next. For example, the gene for eye color is located on chromosome 15. Variations in genes, called alleles, account for the diversity of traits within a population. For instance, there are different alleles for eye color, resulting in brown, blue, green, or other eye colors. Section 6. DNA technology, unlocking the secrets of life. Advances in DNA technology have revolutionized our understanding of life and opened up new possibilities in medicine, agriculture, and forensics. DNA sequencing, for example, allows us to determine the precise order of bases in a DNA molecule. This technology has enabled scientists to map the entire human genome, identify genes associated with diseases, and develop personalized medicine approaches. Genetic engineering is another powerful tool that allows scientists to manipulate DNA sequences. This technology has led to the development of genetically modified crops with improved yields, disease resistance, and nutritional content. Section 7. Medical marvels, DNA and healthcare. DNA plays a crucial role in modern medicine, from diagnosing genetic disorders to developing new therapies. Genetic testing can identify individuals who are at risk for certain diseases, allowing for early intervention and preventive measures. Gene therapy holds the promise of curing genetic diseases by replacing faulty genes with healthy ones. While still in its early stages, gene therapy has shown potential in treating diseases like cystic fibrosis and sickle cell anemia. DNA profiling, also known as DNA fingerprinting, is a technique used in forensic science to identify individuals based on their unique DNA sequences. It has proven invaluable in solving crimes and identifying missing persons. Section 8. Ethical considerations navigating the DNA frontier. As our understanding of DNA and our ability to manipulate it continue to grow, it's essential to consider the ethical implications of these advancements. Genetic testing, for example, raises concerns about privacy, discrimination, and the potential for psychological harm. Gene editing technologies, while promising, also raise ethical questions about designer babies, unintended consequences, and the potential for exacerbating social inequalities. It's crucial to engage in thoughtful discussions and establish ethical guidelines to ensure that DNA technology is used responsibly and for the benefit of humanity. Section 9. Conclusion: DNA's enduring legacy. DNA, the blueprint of life, is a testament to the elegance and complexity of the natural world. From its elegant double helix structure to its role in protein synthesis and inheritance, DNA underpins the diversity and continuity of life on Earth. Advances in DNA technology have revolutionized our understanding of life and opened up new frontiers in medicine, agriculture, and beyond. As we continue to explore the mysteries of DNA, we gain a deeper appreciation for the interconnectedness of all living things and our responsibility to use this knowledge wisely.